My name is Emma Houghton, and I'm here today to present my research Ticks on Lizards, Parasite-Host Interactions of the Southern Alligator Lizard Elgaria multicarinata in Washington State. Ectoparasites are parasites that feed on the outside of the body, such as ticks and mites. Ticks are capable of spreading diseases such as the Lyme disease spirochete. Western fence and southern alligator lizards, which host the disease-causing ticks, have been also found to be capable of cleansing the tick's gut microbiome of the Lyme disease spirochete, which prevents them from passing on the disease to another host, such as humans. Areas where these lizards have been found, such as along the west coast, have often been found to have a lower incidence of Lyme disease. However, southern alligator lizards have not been as studied as western fence lizards. So I asked the questions, what is the general ectoparasite load on the southern alligator lizard population at the study site, and do ticks have a measurable effect on their lizard hosts? I predicted that lizards would show variation in their parasite load because ticks have been shown to often have host preferences for certain characteristics such as sex, size, and so on. I also predicted that lizards with higher parasite loads would show reduced fitness, which has been seen in many other studies of tick-lizard interactions. The figure 1 on the left shows an example of the study site at Catherine Creek near Lyle, Washington, and the figure on the right shows an adult male southern alligator lizard with its slightly larger head than a female, and juvenile on the right side. Lizards were marked, weighed, measured, and sexed before being returned to their capture location. Weight and mass were used to derive a body condition index for each lizard, and any parasites were removed and placed in microcentrifuge tubes for later analysis. I also caught a couple western fence lizards for comparison purposes. All statistical analyses were done in RStudio, and a generalized linear model was used to analyze the data. Unfortunately, despite the many hours in the field, only 17 southern alligator lizards and 6 western fence lizards were able to be captured. However, we saw that males were significantly larger than females. Ticks were found in similar numbers in both lizard species, and snout vent length and tail length may be associated with a slightly increased probability of having ticks. There was no association between weight and the probability of ticks attached. And figure 8 is a good example of a juvenile lizard with an engorged tick embedded on its neck. Sample size was limited previously due to a late start to the field season and a dense poison oak covering the terrain, which made capture difficult. Initial results suggest parasite load does vary among lizards at the study site, supporting my initial hypothesis, and lizards with higher parasite loads do not show reduced fitness, rejecting my hypothesis. Ticks may also prefer lizards with longer tails and snout vent length, which would make sense as it would allow them a better opportunity to attach to the lizard. Results are limited and possibly skewed due to the small sample size utilized. However, significance will be confirmed when research commences after the COVID-19 quarantine. An analysis of tick species and diagnosis of tick-borne pathogens will also be conducted at this time. I want to thank my mentors, Dr. Daniel Beck and Dr. Gabrielle Stryker, for their guidance throughout this project, as well as Dr. Allison Scoville for her assistance with data analysis in RStudio.